Okay, hi everyone. Before we start with today's lecture, I wanted to say congratulations for getting through your first exam last week. The class did very well. The overall average was 84%. I'm very pleased with that because, a lot of those topics are not easy things to grasp. If for those of you who didn't do as well as you would have hoped on that exam, don't be dismayed or discouraged because there's still plenty of time in the semester to kind of revamp and consider different strategies for how to study for this class. I welcome you to email me if you would like to talk about your situation. I'm happy to talk about different ways of approaching the lecture material, how to use the study guides, maybe how you can try different things to overcome some of the challenges you're having in this class. But that kind of depends on you reaching out to me and letting me know that you would like to talk. A couple of you have already emailed me today about that. I haven't had a chance to respond yet, but I will. Okay, so keep that in mind. And of course, if you are concerned about how you're doing in this class so far, it's way better for you and I to talk about this now rather than waiting a few more weeks. You get even deeper into the semester and find it harder to kind of improve your grade. All right, so let's keep that in mind. So one thing that I mentioned to you before is that our textbook tends to kind of go back and forth between discussions of B cells and T cells. So in the two chapters that led up to our first exam, we had sort of introduced B cells, sort of had a sense of how they function, that they have to make immunoglobulins. Then we kind of looked at T cells and looked at how the T cell receptor is made via VDJ recombination also, but the T cell receptor functions different than immunoglobulins, and therefore B cells and T cells have different roles. Right? Remember, B cells make immunoglobulins that ultimately, hopefully, a B cell wants to be chosen to be a plasma cell to make antibodies, right? And then no matter where that B cell is in the body, the antibodies that they make can float around in your blood. Because in order for the antibodies to do their job, the B cell doesn't have to actually be at the site of infection. Right? Whereas we learned with T cells, it's different because the T cell receptor is always stuck to the surface of a T cell. That means the T cell always has to be where the cell that it's going to interact with is. And that's how the T cell knows what cell is presenting something to its T cell receptor that it wants to respond to. Right, so very different functions. So now what we're going to do is we're going to go back and take a closer look at the developmental processes behind how B cells are forming in the bone marrow, and then we'll talk about how T cells are forming. And yes, this does mean we'll revisit the VDJ recombination again, but in this case, we're going to look at how the VDJ recombination is happening in the context of a cell in your bone marrow starting off as a stem cell and kind of becoming a mature B cell, or as we talk about later, a T cell. So let's refresh our memory here and recall that immunoglobulins, right, that are produced by B cells, we can think of these as kind of tickets in sort of like an antigen lottery, right? Now, here in Hawaii, we don't have the lottery, although I did see on the news they're talking about potentially starting the lottery here. But many places on the mainland, including California, where I lived for many years, plays the lottery and has like Mega Millions or Super Lotto. I forgot exactly what it's called. But the premise of the lottery is you go and you buy like a $1 ticket, you pick some numbers, and if you match all 5 of the numbers that are chosen by these little ping pong balls, right, that come out of the little spinning wheel, and you win that, and you match that 6th like mega ball or something like that, then you win a lot of money, right? So I remember when I was living in California, sometimes the lottery would get up to like, I don't know, 20 million or $30 million, but I'd always be like, yeah, that's that's nothing, right? But then once it gets to like, 100 or $200 million, that's when I would pay attention, right? And then I'd be like, oh boy, on my way home from work, I better stop at 7-Eleven and buy a lottery ticket, right? Because hey, you're not going to win unless you actually, pony up the dollar to buy a ticket. So of course, I wouldn't just buy one ticket. I'd buy like 20 because somehow, my odds of, 20 out of 300 million to win is somehow better than 1 out of 300 million. But whatever. The principle, though, behind this is that I never ever fully expected that my combination of numbers would really win, right? Because the odds are just astronomical in terms of getting a perfect match. However, I was never surprised if by the end of the night on the news they said, hey, there are 7 lottery winners, from different places in California that all got the same ticket. Why? Because, well, if you have millions of people playing, right, then even though the odds of individually following one person from the 7-Eleven and seeing that they won the ticket is very astronomically small, you are not surprised that somewhere, right, one or more people happen to have the right number. So that's what I mean when I talk about our immunoglobulins are like playing the lottery. Right? Every single day, you have hundreds of millions of naive, brand new B cells coming out of your bone marrow, each on their surface covered with their unique lottery ticket, right? Not in the form of numbers, but of course, in the form of their unique immunoglobulin antigen binding affinity arms, right, on their IGs, and then on the surface of the B cell, it's covered with that particular IG. Right? Importantly, just like the lottery, you don't have to get a perfect match to be considered a winner, right? Because we're just dealing with immunoglobulins as being close enough matches to a particular antigen that's in your body from something like a pathogen to be good enough that you want to select that B cell and encourage that B cell to divide, make more of itself, and importantly, give you the ability now to make things like antibodies that have this particular correct shape. So just like here, this table here, all right, that shows these are sort of like the odds of getting, let's say, all 5 of your numbers matching plus the mega ball, right? That was like 1 in, I don't know, 258 million or something like that. I mean, that's the one that's awesome. That's when you win like $300 million. And then after taxes, you take home like, I don't know what it is, like 15,000 or something. But anyhow, the important thing here is you don't have to match all of them, right? Let's say you ended up matching 4, just 4. You still get $500. That's pretty good, right? Or you match like 3 and you get $5, something like that. Right? So close enough still gets you some prize money. And that's the thing with the immunoglobulins as well, right? Because all along when we've been talking about choosing or selecting a B cell because its cookie recipe, its particular immunoglobulin shape is the right one, the close enough match, we are not talking about one B cell. We are talking about a population of B cells, all of them having slightly different immunoglobulins that by chance all are kind of close enough to be considered some degree of a winner. Now, that's why, right, we say that our adaptive immune system is so inefficient and expensive. Because what you're basically saying is every day you're making hundreds of millions of B cells, the vast majority of which are never going to match anything or get chosen to match anything. Right? So why did we evolve this very woefully inefficient way of protecting ourselves, through this adaptive system? It's because even though this approach is super expensive, right, the price of failure is too great. Right? Because if you cannot do this and you cannot come up with a selection of B cells to make those antibodies to protect you against something brand new like bad clown, you die. Right? So that's why I think it's interesting to think about that evolution. Normally, we talk about how evolution is all about getting things more efficient, right? That's why, for example, as we evolve, we don't need our appendix anymore. So we no longer have a functional appendix. It's just kind of there. In a few thousand years, we probably won't even have an appendix anymore, right? Or things like, body hair, right? That's why we end up developing things like baldness and things like that. We just don't need it anymore as much. But our immune system, especially our adaptive immune system, is blatantly opposite of that, right? Because we evolved to have a very inefficient, very wasteful kind of approach to doing this, because you have no other choice. It's either that or you die when bad clown comes and infects you, right? So another interesting thing about this is that with the chosen B cells that have close enough fit, yeah, maybe if you're lucky, you do have one that has the 5 winning numbers plus the megaball in terms of like the really close to perfect fit between its immunoglobulin and the antigen from the virus or bacteria. But many of them will be like the matching of the 4 numbers, right? Or matching of 5 numbers, okay? The nice thing about those B cells is they can still be chosen and they can still participate in hypermutation to try to improve that fit. So close enough lottery tickets, in this case, still win some prize money, right? Just like close enough Ig carrying B cells can hypermutate to improve their fit. Unfortunately, I wish you could do that with your lottery ticket, right? You said, hey, I matched 4 numbers, but let me just pretend like I had matched this number and this number. Hey, next thing you know, I should have won 300 million bucks, right? Instead of 500. Okay, too bad we can't do that with lottery tickets, but you can do that with your B cells. This is your bone marrow. This is your red bone marrow found in the long bones of your body, like the long bones of the arm, legs, and also your pelvis, right? So this is the primary hematopoietic site of tissue in your body. Hematopoietic meaning what? Gives rise to all the different types of cells that are in your blood. So in this image here, you can see that in this cross-section of bone marrow, you can make out certain features, like you see some red blood cells here floating around. These big white spots are actually adipocytes. So these are fat deposits. I think you're all sort of aware that bone marrow is very fatty, right? There's a lot of fat in there. But all these other dark purple spots are the nuclei of different developing blood cells. So you have among them things like lymphocytes. I can't really tell which ones are lymphocytes in here, but you also have large cells like this. Perhaps this is a megakaryocyte that's going to go ahead and form platelets, for example. Some of them, you can see they stain a little bit darker. There may be some evidence of sort of granular kind of textures in them. Maybe these are things like eosinophils, basophils, etc. Right. So your bone marrow is a primary hematopoietic tissue. That means primary means that's where these red bloods, that's where these blood cells are born. That's where they come from. So this is where, B cells and T cells come from also. But as we will learn, B cells finish becoming B cells in this bone marrow before they're released in your blood. T cells have to take a little pit stop, a little detour after released from the bone marrow in order to become actual T cells. So let's talk about B cells first today, and then next lecture or the next one we'll talk about T cells. As you start off with a stem cell in your bone marrow, that stem cell is going to divide, and every time it divides, it's going to change a little bit. It's going to differentiate a little bit more, getting closer and closer to becoming something like a full-fledged B cell. Right? Keep in mind that as a stem cell divides, one of its daughter cells is the one that then keeps dividing to provide more and more differentiated sort of intermediate cells that eventually, as they're dividing, form a complete, mature, differentiated cell. Whereas that original stem cell, when it divided, right, one daughter cell started down this path, the other daughter cell hangs around, and it still retains its stem cell-like property. That's why you don't use up your stem cells, right? Your stem cells always hang around, generating more new cells. So what this table is in this slide here is it's an outline. of B cell development starting from a stem cell. And what it's doing is it's helping us frame or understand some key critical changes that take place as part of B cell development. Not surprisingly, because, hey, remember, the whole point of a B cell is to successfully make immunoglobulin on its surface. If it can't do that, it's not a B cell. So not surprisingly, the different stages of VDJ recombination that are part of making the immunoglobulin help define these different sort of phases that the developing B cell goes through. So the way you look at this table is the columns here described sort of what phase or what stage of the B cell development you're looking at. And then the rows going across here are helping to remind you what the status is of the genes in the IG locus on chromosomes 2, 22, and 14 that are coding for things like the heavy chain, so the heavy chain genes or the light chain genes, and whether or not you actually have any immunoglobulin yet, right? So immunoglobulin status. So when you start off with a stem cell in your bone marrow, right? And both the H chain genes and the L chain genes, right? Because nothing has happened yet. So this is a reminder that the stem cell, the version of your immunoglobulin locus that you have with all the V blocks, D blocks, and J blocks, it's whatever mom and dad gave you, right? That's what you inherited through the germ line. So the stem cells always maintain the sort of germline status of all your different colored blocks available for you to begin doing VDJ recombination. And then obviously the stem cell does not have immunoglobulins on its surface. So that's why the IG status here is none. The three main stages that a developing B cell goes through is the pro-B cell stage, the pre-B cell stage, ending up with an immature but sort of ready B cell at the end. Okay. The pro-B cell stage is mainly preoccupied with rearranging the heavy chain to try to make a heavy chain, a usable heavy chain gene that can then create the heavy chain part of the immunoglobulin. Right? That kind of goes into the pre-B cell stage, but then the pre-B cell stage, once the heavy chain is made, now you're looking at trying to make the light chains. Right? So you can see here, I've broken this down using these brackets. The pro-B cell stage going into the pre-B cell stage is working on making the heavy chain of the IG, right? Whereas the latter half of the pre-B cell stage, finishing up with you having an immature B cell at the end, is working on the IG light chains, right? This one and this one, right? If you think about it that way, and then you look at this table, you realize it's not that confusing because it's helping you remember the status of what's going on at the heavy chain locus and the light chain locus, right? And where the VDJ steps are taking place. So it's not so much about me expecting you to understand or filling in this table, because in some ways that's kind of arbitrary, right? The author of the book decided to organize it this way with these particular comments in each one. Really what I want you to understand is, why is there a pro-B cell stage? Why is there a pre-B cell stage? What the overall differences are in terms of what the developing B cell is going through during these different stages. All right, so let's start off with the pro-B cell stage. So this is primarily when the heavy chain locus gets rearranged. Remember that VDJ recombination takes place when you have all these different available fragments to choose from, the colored blocks, the V's, the D's, and the J's, and a particular developing B cell will make its own sort of decisions, sort of at random, as to which colored blocks to try to put together, representing different fragments of a gene that, if they're put together properly, form one usable, transcribable, and translatable gene sequence. Right? And so that's done by our friend, the RAG recombinase complex, the oven mitt. Right? So we already learned that that the end of one of the immunoglobulin arms, right, so the tips of my fingers here, that's the three-dimensional surface that is variable from one IG to another IG, if you're looking at two different B cells, right? And that's where the money is, because that's the actual three-dimensional surface that you hope will pay off and match a three-dimensional epitope presented by an antigen. Right? So we said that those surfaces utilize these highly variable loops, right, these highly variable domains, and we said that 2 out of the 3 loops come from just your choice of which V block that you choose. And then the third loop is a product of that random kind of process of linking together the cut rope, right, or the cut DNA that has to be joined back together again. Right? One thing that we didn't talk about earlier, but it may occur to you, is this random addition of DNA bases, right, that creates the variability in this HV3 region. You know, that has a very good chance of messing up the reading frame of the resulting gene that gets put together. Remember, reading frame means our DNA, when it's transcribed into RNA, every 3 nucleotides is read as a codon, right, that specifies the next amino acid that needs to be put together to make a functional protein at the end. Right? So if you mess up the reading frame, like you have an uneven number of nucleotides, so instead of like 3, 3, 3, the actual process of joining through VDJ recombination, right, you have a number of nucleotides that aren't a multiple of 3, then everything can shift in terms of how the genetic sequence gets read. So that means that as these blocks are coming together, you may actually mess up the reading frame, and the joining of these different blocks may end up being useless, right? Because then later when the cell tries to make a protein from this, it's garbage. Right? Interestingly, in the case of VDJ recombination with the heavy chain of IGs, the D and the J blocks always end up joining together in a way that you still get a sensible amino acid sequence. Part of that is simply because the D and the J blocks are actually quite a bit smaller or shorter than the big red V blocks. All right, so even if you get a little frame shift in there, the resulting amino acid differences that you get from that frame shift, right, can still successfully be part of the finished heavy chain. However, the joining of the V block to the D block, this is more critical. So you only have a 1 in 3 chance of getting these 2 blocks to join up in a multiple of 3, right? So that you actually still have usable codons that make sense, and that you don't end up with the amino acids being wrong, you know, or ending up with some sort of like a stop codon or something like that. So we say you have a 1 in 3 chance of making a productive rearrangement here. Right? In other words, you have a 2 out of 3 chance of getting these things to join up in a way where you've messed up the reading frame and it's no longer usable as a gene. Okay. So our B cells, once they start down the path of becoming a B cell, and they say, okay, I got to try to do this VDJ recombination and make an immunoglobulin here, right? It starts with making the heavy chain. Evolutionarily, that makes sense, because remember, the heavy chain involves choosing 3 different blocks: a V block, a D block, and a J block, right? So that's harder to do than the light chain, which doesn't have a green D block. So all you're doing is combining a V and a J. So the cell, evolutionarily, might first see if it can successfully make a heavy chain first, right? Because that's the harder one to do. And then it'll worry about the light chain afterwards. So during the early pro-B cell stage, it will make a productive rearrangement of a D block and a J block. Keep in mind that you have 2 heavy chain loci in your cells, one that came from mom and one that came from dad, right? So when this starts off, both of those loci are doing VDJ recombination. So the choice of a D, a choice of a J, and it's not the same on both, because remember the oven mitt, or oven mitts, because there's lots of oven mitts floating around, right? There's nothing that says it has to be the same decision on both chromosomes as to which one you make. Right, so that's why this first DJ, hopefully that works out. It's always a productive rearrangement. So the way you look at these flow charts is that if you have a productive rearrangement, or the joining of these 2 chosen blocks, there's a plus, right? So it's productive rearrangement. If it's non-productive and you screwed it up, it's a minus, right? Negative. After D and J gets recombined, so now you move to this side over here, you're now into the late pro-B cell stage, right? Still, you're preoccupied with rearranging this heavy chain to get it to work. So here you've made the D and the J, right? This is what you made from the left over here, right? But now you're showing it, that part's been made. Now you choose a V block and try to join it up. And so this is where the sort of 1 and 3 comes in, right? So on one of the chromosomes, either the one that you got from mom or dad, let's just say you start trying to make that rearrangement. So your RAG protein oven mitts in the B cell chooses a V block and tries to add it to the DJ that's already been put together, right? Oh boy, maybe that didn't work, right? So if that didn't work and you screwed it up, well, you kind of have to say, forget about that first chromosome. But fortunately, we have another one, right? If we started with the one from dad and we messed it up, now you can try to do it on the one from mom, right? And you try to do that same thing. If that doesn't work out, oh boy, because remember, it's only got a 1 in 3 chance of it working out, then that's the end of that B cell, right? I mean, it never even became a B cell, but the whole point is this particular cell that was trying to be a B cell, it didn't succeed, right? Because it basically used up its ability to make a heavy chain. And so about half of all developing B cells just stop at this point. Wow, you talk about wasteful, right? So not only are you making hundreds of millions of B cells every day, the vast majority of which never amount to anything because they're not useful. If you go back inside the bone marrow, right, those hundreds of millions that even made it out of the bone marrow only represent 50% of the B cells that started down that pathway to try to become a B cell. However, if at either of these steps you succeed in properly getting a V block joined up productively with a DJ block, right? So about 50% of the cells succeed in doing that and move on now out of the pro-B cell stage into the pre-B cell stage. So at this point, the developing B cell will make the heavy chain protein from this and evaluate whether or not this heavy chain protein actually looks right. How does it do that? Well, much in the same way that, let's say, a carpenter or a woodworker who's making, let's say, a whole bunch of tables, right, and wants to make the same kind of table, what the woodworker may do is, as he or she is shaping different things like the corners, he may have some sort of little template. So in this case, he has this piece of wood that he knows is exactly the right angle that he's kind of shooting for this particular joint. So after he's putting this joint together, right, he kind of fits this to make sure, yep, that's a good angle. That's the one that I need, right? And this way he can very efficiently make a whole bunch of these different corners for the table. So the developing B cell does a very similar thing. Here you see on the left, the developing B cell has successfully done its VDJ recombination to make its heavy chain. So now it makes the product of this VDJ choices, right? It transcribes it and then translates it. And now you have this heavy chain, which is this dark blue, heavy chain here. Right? So inside of the cell, this is inside of a vesicle inside of the cell. That's why you can tell it's kind of, on the wall of this vesicle here. The B cell has these other proteins, this V pre-B and lambda 5, right? That essentially are working to check the shape of this heavy chain to make sure it is the right shape, just like these would be the little piece of wood here that the woodworker is using to make sure that the corner of the pieces that he's working on are the right angle, the right shape. So these V pre-B and lambda 5 proteins, they basically come together and kind of form like a fake light chain in a way, right? So they are the right shape to kind of fit along the heavy chain. And if they fit nicely, right, it's like a little template, a little mold that the B cell is seeing if these fit, then they will be put into the right conformation where they will then signal the B cell that, hey, this looks good. This heavy chain that we came up with, it sure looks like a heavy chain. It's the right size, it's the right shape, etc. Right? So it's like a quality check. If these V pre-B and lambda 5 proteins don't successfully bind to the heavy chain, right, then the cell never gets the signal to continue on. And at that point, that B cell, that's the end of it. It's not going to go any further. So that's why as we go into the pre-B cell stage, you can see the status of the heavy chain is that the V, D, and J gene fragments have been rearranged now. Nothing has happened yet in the light chain. That's why the status of the light chain is still germline. But in terms of the Ig status, well, yeah, you now have sort of part of an Ig made. You have the heavy chain, right? And remember, the very first Ig that's made by a developing B cell is the mu constant region, stem type or isotype of Ig, right? So these mu are these constant blocks that are chosen to go along with the VDJ arranged red part here. Right? Then as you move through the pre-B cell stage, now the cell starts rearranging the VJ blocks at the light chain loci. Right? And it's going to work on the light chain loci now. Let's take a look at what's happening there. So the light chain formation is more efficient than the heavy chain formation because, again, the heavy chain, you had to succeed in properly joining 3 different gene fragments together, whereas with the light chain, you just have to succeed in joining 2, right? A V and a J, a red and a yellow. So let's look at this kind of scenario here. So you have several V blocks to choose from. Right? As we learned, there's maybe like 30, 35 different red blocks to choose from. And in this case, the cartoon is showing 5 different J blocks to choose from. Let's say that in this particular developing B cell, the oven mitt chooses variable block kappa 1, right? Because this is the kappa light chain loci. Remember, right? Even though you have 1 heavy chain loci in the human genome has 2 light chain loci, a kappa and a lambda on different chromosomes. So you start with the kappa one here. The oven mitt chooses variable block 1 and J block, I guess this would be number 2 over here, right? And joins them together. Oh, when these came together, it wasn't productive, right? The reading frame got messed up when the DNA was stitched together because the number of bases that was randomly used is not a multiple of 3. Right. So fortunately, already the B cell has put a lot of work into this, right? It already went through the whole making a heavy chain. It's kind of a shame if the B cell just calls it quits and poops out at this point. You have other blocks to choose from. So if that first VJ recombination is non-productive, as long as you have other V blocks and J blocks to choose from, the oven mitt can try again, right? So now it chooses the variable block 2 over here, and yellow block, J block number, I don't know what this was. This was number 4 before, right? It tries that, a second VJ recombination, right? Ooh, that also screwed up and it didn't work, right? Because the odds are against you. It's a 1 in 3 chance of the reading frame being preserved. Fortunately, right, there's still more red blocks and more yellow blocks to choose from. The rag oven mitt tries one more time. It chooses this V block, and in this case, the 5th of the yellow J blocks, right? And this time it succeeded, right? So, oh, good. After 3 rounds of VJ recombination, it did work, right? Good thing it did, because it's not going to go any further than that, because in this illustration, this cartoon shows it's used up all the available yellow blocks, right? So again, remember, depending upon which colored blocks the oven mitt chooses on the big piece of rope, it might be by chance that it chose a red and a yellow block that already was kind of close together, right? So the part of the loop, part of the rope that gets looped out really doesn't contain many red blocks or J blocks that are lost, right? In which case that would be like this, because if that didn't work, you still have more red and red and yellow blocks to choose from. But if the oven mitt had started by choosing, let's say, the very first red block and the very last yellow block, then all the colored blocks in between would have been lost on the loop of rope, right? And in that case, that developing B cell, if that VJ recombination that it's trying doesn't work, it's out of luck, right? And that B cell would have to just terminate and it wouldn't go any further. All right. So that's what's illustrated here. Again, just like when we looked at the flow chart before. So in the pre-B cell, it's going to do its light chain rearrangement here. It's going to try rearranging one of the kappa light chain loci on whichever is the first chromosome, mom's or dad's, right? If that doesn't work, hey, it's okay. You can try it again on the second chromosome, again, mom or dad's, whichever one you didn't use first. And if that works, awesome, right? You now have a light chain that's made. And importantly, this is the kappa light chain, because that's the one that you're trying to make first, right? Then you're in business. These green kappa light chains now help assemble with the existing heavy chain that you made, and you have your IG. But what if, things don't go that well? Let's say you screw up a couple times here. You've used up both kappa light chain loci on your 2 chromosomes, right? It's okay, because evolutionarily we have that other light chain loci also, or locus also. So we have the lambda light chain. So again, maybe you start from mom's first. Ooh, that didn't work out. Then you try to do dad's. Yay, that worked. Okay, so now you end up with this other form of the IG that has these orange lambda light chains. And I think I told you before, to this date, we're not sure functionally if there's any difference between this IgM that has the green light chains or this IgM that has the orange light chains. So this one is the kappa light chains, this one is the lambda one. Functionally, we're not sure if there's any difference. It's quite possible that we just evolved this way because, again, it's a terrible shame if our B cells went through all that work getting to this point just to kind of lose out and not be able to successfully make an IG, right? So maybe that's why we evolved 2 chances to make light chains from 2 different loci. But hey, some of the cells aren't going to succeed. And then those cells, right, if they fail to make either of the types of possible light chains, they undergo apoptosis, and that's the end, and contributes more to the wastefulness of this whole process, right? So at the end, if you do get an IgM that's formed on the surface of the B cell, that's what we call our immature B cell, right? And you say, why is it immature? Because I thought the whole point was now that it has an IgM on its surface, or more specifically, its surface is coated with the IgM that it's made. I mean, isn't that a B cell? No, because it still has to go through another very critical set of checks. Okay, what does that have to do with? Okay. Let's look at this. This is a summary, right, of what we've been talking about. So it's another nice way of visualizing this. The early pro-B cell, it starts trying to make its heavy chain. So in that early pro-B cell, it starts making these little template proteins. These are the little templates like the woodworker would use to sort of quality control things. So the cell has these proteins made, like the V pre-B and the lambda 5, ready to check out any heavy chain that's made, right? So the first checkpoint would be, right, you've made this form of the receptor or of the Ig that's not a full Ig yet, because this is just 2 heavy chains together. So we call this form the pre-B cell receptor, right? If the V pre-B and lambda 5 confirm that this is the right shape, then you move on, right? If the V pre-B and lambda 5, don't detect that there is a proper heavy chain pre-B cell receptor made, or the shape of it is wrong. And this cartoon is just showing that, like, the cell couldn't even make it, right? Because the rearrangements were non-productive. Then that's it. The cell just dies here, right? But moving on, if you have the pre-B cell receptor made of just heavy chains, then the second checkpoint would be you try to make the light chains, right? And again, if you can't do it and the light chains don't appear, right, then the cell undergoes apoptosis. Right? But if it does work and you assemble a functional IgM, now you have your immature B cell at the end of this process. Right. So I said, why is it still considered immature? What is this other super important sort of checkpoint that it has to go through? This has to do with the fact that this whole creation of a 3-dimensional shape for the antigen binding domain of the new immunoglobulin that this new B cell has made has been more or less random, right? There's so much variability, and that's how it's designed, to have the utmost variation and variability. Several of you have asked me in this semester so far, I don't understand it, Dr. Shen. If these are all random shapes, then aren't some of them going to be shapes that are actually already present in our body, just from our body being filled with cells and proteins and carbohydrates and all kinds of molecules? Yes, right? In fact, many of them will be the right shape that matches this brand new immunoglobulin on this B cell. And if that's the case, you do not want this B cell to be allowed to go to work in your immune system, right? Because this B cell will probably end up detecting, right, some antigen in your body that is a totally normal, healthy antigen that's already present. And if that happened, right, your immune system would auto-react. So that's why this last part of the B cell's development is so important, because it has to do with the careful handling of recognizing self, right, versus non-self. So in the case of a B cell's immunoglobulin, let's say you have a B cell here that was allowed, or this is an Ig that came from a B cell that was allowed to go to work in your immune system. And here, let's say this is a peptide or some other normal molecule in your body, a protein, a carbohydrate, some sort of lipid that just happens to have the right epitope shape for this antibody to bind to. That would be trouble, right? Likewise, as we will learn later, our T cells also have to go through this important consideration, right? Because here you have a T cell that is unfortunately the right shape to bind to totally normal self-peptide that's coming from one of your normal healthy body cells that's just presenting that self-peptide on its surface, like the cells normally do, right? So it's easy to see why such self-recognition is bad, right? And you don't want B cells or T cells that do this to be allowed to go to work. But there are some situations where recognition of self actually is a good thing, right? In particular, with the development of certain types of diseases from within your own body, like cancer. Okay. So disease cells, like cancerous cells, may actually express normal peptides, but just in outrageously out of whack proportions, right? Way too much of certain proteins. In that case, those can become useful antigenic signals for your immune system to target your own diseased cells and use your immune system to eliminate them. We will talk about cancer later on in the semester. All right. So then how does this B cell, since we were talking about B cells today, how does it go through this final kind of evaluation as to whether or not it's going to be causing trouble, right, if it's allowed to go into the body? So when it's still in the bone marrow before it's released, here you have your immature B cell that successfully made its IgM, right? All that laborious process of making heavy chain, light chain, etc. This particular cell managed to do it, right? Unlike many of its little brothers and sister cells that couldn't handle it and died along the way, right? So the final test is, does this IgM on its surface actually happen to be, due to bad luck, the right shape to bind some self-antigen, right? So in the bone marrow, it's surrounded by examples of self-antigens. Other cells that it's interacting with in the bone marrow tissue present a whole bunch of normal three-dimensional molecular shapes, right? So let's say here's a bone marrow cell in the bone marrow with the B cell, and it has on its surface this triangular shaped molecule, right? And it turns out that, it turns out that this IgM, the little ends of it, right, you can see they're sort of flat, they're not triangular shaped, they don't bind, right? So it doesn't encounter anything in the bone marrow that causes trouble. Therefore, this immature B cell is allowed to move out into the blood, right, and now start taking a look at whether there are other potential self-antigen signals that it might bind to in the blood. Right. What does that suggest in terms of how the IgM on the surface of the B cell signals at this point? Right? Because normally we think that Ig on the surface of a B cell, if something binds to it, that would actually send some sort of stimulatory signal into the B cell's nucleus that would sort of like tell the B cell it's been selected or chosen, right? And that B cell starts dividing, and then might await further instructions to become a plasma cell or hypermutate or whatever, right? But in this case, at this stage of the B cell's development, right, the wiring, so to speak, of its IgM is backwards. If anything binds to the IgM at this point, right, the assumption is that thing binding to it is actually a self signal. So the IgM will actually negatively signal the nucleus of the B cell, right, and tell the B cell, uh-oh, right, we have a problem here. Okay. So that would happen both in the bone marrow as well as after the B cell is released into the blood. But let's look at what happens if you have this B cell over here that is also finishing its development in the bone marrow. It reached the immature B cell stage, but look at its unique Ig. Oh, it has the little triangular shaped antigen binding domains, right? So it interacts with self signals that are present in the bone marrow. And again, that sends negative signals into the B cell, right? If it turns out that this B cell did not use up all of its V and J blocks in making its light chains, right, this B cell has a second chance. So it can still do VJ recombination again, and the oven mitt will try to choose a different red block, a different yellow block, recombine those to see if that little change is enough, right, to get it so that this B cell doesn't have the right shape anymore to bind the self-antigen signal. All right. It can still do this because it's in the bone marrow, so it's still in the environment where the VDJ recombination is happening, right? And importantly, it still has extra or spare colored blocks to choose from. Right? If the B cell is out of the bone marrow, or if the B cell has already used up all of its possible V and J blocks, then that's the end of the line for this B cell. Okay. So this will be the last slide for today. I think I'll stop here. So this slide kind of summarizes then what we've been talking about so far in the developmental pathway of B cells. So the developmental pathway of B cells involves two sort of main sort of locations within your body. The first few developmental phases, the ones that we've talked about today, are these sort of yellow ones, right? And they take place in primary lymphoid tissue, like the bone marrow of the long bones of your arms, the legs, and also the bones of your pelvis, right? So that has to do with assembling through VDJ recombination a good Ig, immunoglobulin on its surface, right? And then you're going to have to select, negatively select for those immature B cells that improperly auto-react or end up detecting self-antigens, right? A fraction of those will actually be okay. And these will then be allowed to go out of the bone marrow into the blood, right? So here, naive B cells, remember, a naive B cell is a B cell that has never actually had it bound yet to the particular epitope shape that its Ig is designed to bind to, right? So during the process of development, you might have a B cell that accidentally binds to self-antigen, in which case maybe it has a chance to try again, right? So the actual immature B cell that you end up with has something that doesn't bind to self-antigen, so we can still technically call that version of it naive, right? Because whatever it's destined to bind to, it has not had a chance to see it yet. Right, so naive B cells are made in the bone marrow, and a reminder that's a primary lymphoid tissue. When we continue our discussion next time, we'll look at once it's released into the blood and it finishes this negative selection process in the blood, it's going to make its way to secondary lymphoid tissues. Secondary lymphoid tissues such as our lymph nodes, our spleen, or particular types of immune tissues in our intestines known as Peyer's patches, where the B cells can actually mature, right, and become properly readied to really go to work and start looking for pathogenic antigens. Okay. All right, so we'll stop there today. There's one more slide, but I'll save that for our Friday lecture. Okay. So let me stop the recording.